Friends, in this module of PCR failures and case studies, I shall discuss about what is polymerase chain reaction, what is PCR technology, common reason of PCR failures, solution for PCR inhibition and case studies related to PCR failures. Polymerase chain reaction, in short PCR, has proved to be of great importance to forensic DNA typing laboratories. Carey Mullis and members of the human genetic group at Roche Molecular System US first described this technique in 1985. Due to the ability of PCR to make millions of copies of particular DNA sequence in just few hours has revolutionized molecular biology. DNA amplification with PCR technology is sensitive, rapid and is not limited by DNA quality like other techniques such as RFLP, that is restriction fragment length polymorphism. It is well suited for forensic DNA sample analysis. Now, what are PCR errors? What errors take place while carrying out PCR? Polymerase chain reaction is not always free from errors. There are many factors which cause error and inaccuracies in PCR products. DNA polymerases are used in PCR reactions. Most commonly Taq DNA polymerase is used. These DNA polymerases do not have the ability of proof reading, which results in error in amplification. Probability of occurrence of errors is more in case of greater amplification. Sometimes the product are formed from non-target site leading to mispriming, which is a major problem. Sometimes one primer is annealed to another and cause primer extension. This leads to formation of primer dimers, which are byproducts of polymerase chain reaction. Formation of primer dimers may produce unexpected variability in PCR success. These all factors may lead to PCR failures. Now, what is chain reaction inhibitions? The presence of inhibitors in DNA sample collected from the crime scene affect the PCR amplification process of DNA. In case of outdoor crimes, sometimes body fluids are found on soil, wood or sand etc., which get mixed with criminals' DNA sample and may prevent PCR amplification. Various other substances such as textiles dye, leather, wood etc., may also contain inhibitors which inhibit DNA polymerase. These inhibitors can affect in following ways. Number 1, during DNA extraction they can interfere with the cell lysis. Cell lysis, which is an important step for DNA extraction. Secondly, they can degrade nucleic acid or capture it, thus interfering in the process. And thirdly, enzymatic amplification of target DNA can be prevented due to inhibition of polymerase activity. Sometimes substances like hemoglobin from red blood cells or textiles dyes remain in DNA sample throughout the sample preparation and then prevent the PCR amplification by interfering with the polymerase. If amplification of DNA is done with inhibitors such as hematin, then it may lead to loss of alleles from large STR loci or even complete failure of all loci. Partial profiles are produced in case of sample containing inhibitors and produce results which are identical to partial profiles produced in case of degraded DNA samples. Therefore, due to presence of inhibitors or due to degraded DNA sample, amplification of large STR loci fails. If samples contain inhibitors, then reduced size STR amplicons are used as they aid in recovery of information from a sample. Amplification of smaller PCR products take place more efficiently as compared to amplification of larger PCR products. Common reason of STR failures are as under. It is usually difficult to find the cause of PCR failure. Causes of PCR failure are complex. Failure can be due to multiple factors or due to only one factor. Proper analysis of all the factors is very important for getting accurate amplification. Following are some of the reasons which lead to PCR failure. Number 1, inadequate DNA template concentration. A correct determination of DNA template concentration is not always possible with spectrophotometry. It rarely gives a true determination due to presence of chromosomal protein and RNA etc. The best way to determine the DNA template concentration is by visualizing the stained DNA on agar gel along with standards of similar size and known concentration. Inadequately purified DNA template. DNA template may also contain carbohydrate, salts and lipids apart from DNA, proteins or RNA. The presence of these substances can inhibit the activity of dark DNA polymerase during PCR. Unwanted contaminants can be removed using purification devices. For cleaning the DNA template significantly, ethanol precipitation should be done after column purification elution. PCR products. Transfer of PCR reaction components to the cycle sequencing reaction due to unpurified PCR products may cause less efficient result. Dilution of vital fluorescent dideoxy terminators take place due to carryover DNTPs. If the PCR reaction is non-purified, then it may contain multiple products which can produce multiple reads due to binding with sequencing primer. Failure to stick clone to single colony due to lack of unexpected primer binding site. This type of failure can occur in the template. Failure to fully characterize a new plasmid construct can result in a template which contain an altered binding site or lack expected primer binding site. This failure can take place even while designing cross species primer pairs. Very less amount of primer may also lead to generation of low amount of extension product showing up low signal and low resolution read. Large amount of primer does not create any problem, but it should be avoided. If the temperature of the primer melting is too low, then less stringent primer is produced during annealing step and the probability of primer binding to other site on the template increases. Secondary base pairing along the DNA molecule restricts or prevents the movement of dark DNA polymerase along the DNA template at the point where a knot or fold is formed. These internal folds, which are formed due to base pairing between nucleotides within a single stranded DNA, are known as hairpin loops. These secondary structures can be opened up if DNA relaxing agent such as betaine or dimethyl sulfoxide is used in cycle sequencing reaction. Overlaying of resultant reads takes place if multiple priming sites are present and the primer binds to more than one site. Sequencing incorrect PCR products may lead to failure of polymerase chain reaction. The product extracted from the agarose is not the target product. Formation of primer dimer is one of the potential reasons for PCR failure. The primer sequence can anneal to itself or to other primer, which results in minimal binding to DNA template. Sample handling error may also lead to failure of polymerase chain reaction. If wrong primer combination of wrong template is used in the cycle sequencing reaction, then it may also lead to PCR failure. Now, what are the solutions of this PCR inhibition? For that, proper steps or solution must be undertaken to prevent PCR inhibition. PCR inhibition may lead to polymerase chain reaction failure, producing unreliable and inaccurate results. Following solutions can be used to remove PCR inhibitors or reduce their effect. Number 1, dilution of genomic DNA leads to dilution of PCR inhibitor. After dilution, the sample can be re-amplified in presence of less inhibitor. To overcome the effect of inhibitor, addition of more DNA polymerase can be done. By doing this, some portion of TARC polymerase bind to the inhibitor and remove it from the reaction. The remaining polymerase can do its work and amplify the DNA template. DNA polymerases other than TARC DNA polymerase work better in presence of inhibitor such as blood and fecal matter. To prevent or minimize the inhibition of PCR, certain additives such as bovine serum albumin or betaine are added to polymerase chain reaction. To neutralize inhibitor of TARC polymerase, DNA can be treated with sodium hydroxide. In case of DNA sample collected from soil, ammonium sulfate is added to the sample, which helps in preventing the co-purification of inhibitors along with DNA. At last, a separation step may be performed before polymerase chain reaction in order to separate the extracted DNA from inhibiting substance. Now, I shall talk about some case studies related to PCR failures. Investigation of the effect of DNA degradation and inhibition on PCR amplification of single source and mixed forensic sample. Studies have been conducted by Bruce McCord and others to assess the effect of DNA degradation and inhibition on PCR amplification of single source and mixed forensic samples with the goal of examining the mechanism of PCR inhibition and degradation by performing the following experiment. Number 1, the use of real time PCR to analyze the effect of various inhibitors on PCR amplification. And number 2, analysis of PCR profile for effect of natural and enzymatic degradation. Number 3, analysis of DNA profile for effect of chemical oxidation. And number 4, correlation between DNA amplification and PCR inhibition. Based upon the experiments, it was concluded that DNA in tissue sample becomes almost unrecoverable due to environmental damage and the template breaks down into small pieces. Real time PCR proved to be an effective tool for detection of PCR inhibition. Experiments on effect of DNA template sequence, DNA template length and inhibitor concentration showed that PCR inhibitors may have effect on STR results. TARC polymerase is affected by DNA inhibitors. The total amount of DNA produced is reduced, which leads to loss of available template. The sequence or length of DNA also affects inability to bind. Those inhibitors which affect Taq polymerase inhibit the DNA by affecting the largest alleles first, whereas in case of inhibitors which bind to DNA affect smaller and larger allele both. Small sequences or mini STR must be used to improve resistance to PCR inhibition. Improvement should be made according to type of inhibition. Inhibition which is sequence specific may still cause issues with small amplicon. Next is a study of PCR inhibition mechanism using real time PCR was carried out by Kelly, L. Opel and others. They studied the mechanism of PCR inhibition by examining the effect of melting temperature, amplicon length and sequence. A single homozygous allele was selected and specially designed primers with 3 different amplicon length and 3 different melting temperature was used to target the selected allele. Variable concentration of various inhibitors was added to the reaction mixture to study the effect of PCR efficiency. According to the results, depending upon the type of co-extracted inhibitors, various type of inhibition mechanism can take place during PCR. These inhibition mechanism include Taq inhibition, effect on reaction efficiency and DNA template binding. Some inhibitors like tannic acid affect in more than one manner. Some smaller amplicon were also affected by inhibitors. Thus, the hypothesis that smaller amplicon are more efficient in amplifying inhibited sample is not correct. It was concluded that melting temperature and amplicon size are important in some inhibition mechanism, but not for all. The main issue in understanding PCR inhibition is determination of identity of interfering substances. Kenneth D. Allert and David R. Foran conducted a study on polymerase resistance to polymerase chain reaction inhibitors in bone. The presence of naturally occurring inhibitors in DNA from aged or degraded skeleton remains makes amplification of DNA a challenging task. PCR inhibitors may inactivate polymerase or may bind to other reaction component. According to their study, 10 thermostable polymerases from 6 bacterial species were examined to study the ability to amplify DNA in presence of PCR inhibitors. Lower susceptibility to inhibition from bone was shown by 2 polymerases, one from Thermus thermophils and other from Thermus aquaticus, whereas high susceptibility was shown by polymerases from Thermus flavus. The activity of enzyme was improved on addition of bovine serum albumin. So, according to the result, it was concluded that the thermostable DNA polymerases have different susceptibility to bone derived PCR inhibitors, and those polymerases which are commonly used in forensic laboratories may not be optimal when working with DNA from skeletal remains. Therefore, inhibitors present should be analyzed first and then polymerase for analysis should be chosen to avoid any PCR failure. The whole topic can be summarized in following points. Number 1, polymerase chain reaction is not always free from errors. There are many factors which cause errors and inaccuracies in PCR amplification. There are many factors which cause errors and inaccuracies in the production of the PCR products. DNA polymerases are used in PCR. Most commonly, Taq DNA polymerase is used. These DNA polymerases do not have the ability of proofreading, which results in errors in the amplification. Presence of inhibitors in DNA sample collected from crime scene affect the PCR amplification process of DNA. Partial profile are produced in case of sample containing inhibitors and produce results which are identical to partial profiles produced in case of degraded DNA samples. For cleaning DNA template significantly, ethanol precipitation should be done after column purification elution. Common reason for PCR failure are inadequate DNA template concentration, inadequately purified DNA template, failure to stick clone to single colony, failure to fully characterize a new plasmid construct, calculation error in primer concentration, primer melting temperature too low, secondary structure in DNA template, multiple priming sites when present, sequencing incorrect PCR product, primer dimer formation and sample handling error. PCR inhibition may lead to polymerase chain reaction failure, producing unreliable and inaccurate result. To prevent or minimize inhibition of PCR, certain additives such as bovine serum albumin or betaine are added to polymerase chain reaction.